Hello and welcome to Medical Microbiology. I am Dr. Van Laar and I'll be your instructor this term. Medical microbiology is going to cover all aspects of organisms that cause disease in humans, and we are really going to be looking at the symptoms of these diseases, the treatments of these diseases, and really importantly, how you can differentiate one set of symptoms from another. So throughout the term, we'll be talking about a lot of different organisms that cause a lot of similar types of infections, and your goal is going to be able to pick up those subtle differences that will allow you to determine one causative agent versus another. The course is going to be broken up into bacteria, then viruses, then fungi, then other eukaryotic parasites. So we're going to try to cover a really broad spectrum of organisms that can cause disease, including those that are new, like the SARS coronavirus, COVID-19, that may not be in your textbook, but your textbook is an excellent resource, and any of you considering clinical laboratory science or medicine as a future career should definitely be investing in the book. Because of COVID-19, I am recording all of these lectures from home. Those of you who have had me in other classes already know about the zoo that I have living at my house. So at some point or another, like right now, you may hear toys squeaking, dogs barking, cats meowing, a bird chirping. It is what it is. It's a really different situation. Not every video is going to be like that, but right now, one of my dogs, his name is Barney, is enjoying playing with his toy. So let's go ahead and kick off medical microbiology, discussing how we would diagnose infections. And again, there's going to be a lot of information in this class that should be a rehashing or a review of what you learned in microbiology, especially if you took microbiology with me. So I am operating under the assumption that you know microbiology. If you did not take microbiology, you should not be in this class. If you did not perform well in microbiology, this class might be a big challenge for you. If you did very well in microbiology, you will likely do well in this class as long as you put in that same level of effort. When we're talking about diagnosing infections, there are a number of different mechanisms that health professionals can use to diagnose the causative agent of a patient's disease. One of the most common methods for diagnosing infection is using microscopy. Now, depending on the type of organism that you suspect is causing the infection, you may use different types of microscopy. Very commonly, most labs will have bright field microscopy, which is just the classic microscope that you would have used in any of your laboratory classes. Some will also have dark field microscopy. It is a version of light microscopy that allows you to visualize slightly smaller or thinner organisms. When I was doing my PhD, we used dark field microscopy to visualize spirochetes. Phase contrast microscopy, again, is a variation of light microscopy that is useful for seeing living organisms. So perhaps looking for amoebas in somebody's cerebrospinal fluid. Fluorescent microscopy, another modification of light microscopy that is useful, especially if you're looking for very small organisms or organisms perhaps embedded in tissue. And then electron microscopy. Most labs won't have that. Electron microscopes can be very expensive, upwards of $250,000, but they can be useful for visualizing viral specimens. So light microscopy, as I mentioned, most commonly used, and of course bright field is the most common type of light microscopy. This is what you would see with light microscopy. You'll see a lighter background and the organisms will appear darker. Sometimes, especially with things like bacteria, they are so small and translucent that you'll have to apply different types of stains to the microorganisms in order to visualize them under the microscope. This is the lowest resolution. So remember, when we're talking about microscopy, resolution is the ability to see two separate, let's say we're talking about bacteria, two separate bacteria as two separate bacteria. And so resolution is dependent on the wavelength of light. The longer the wavelength of light, the less resolution you get. So with visualizing very, very small things, we can only use visible light. That's all we can see with our eyes. And so our resolution is going to be limited. This is why you cannot see viral particles using light microscopy, because the viruses are too small and you don't have high enough resolution. So with light microscopy, especially if you use an oil immersion lens, you can often see the majority of bacteria, but that's about it. Dark field microscopy is similar. It still relies on visible light, but in this case, the sample would be light against a dark background. So you see basically the same slide as you see here, except it's visualized with dark field microscopy. So the background is dark and the cells are lit up, the cells are light. And so this is useful, again, for organisms that are very thin, like spirochetes. With these cells, you see that you can also see sometimes a little bit better some of the thin extracellular structures that these cells might have. So that's an advantage of dark field. So it gives you a slightly increased resolution, and like I said, it's used commonly for spirochetes. Phase contrast microscopy allows for imaging of internal structures. So you can see in this eukaryotic organism a lot of the internal structures. It's also useful if the cell is still living. So it gives you a 3D visual of live samples. And the reason that you can see all of these different samples is because the cells and their internal structures all refract or bend light differently. And so that allows your eyes to make a three-dimensional picture. With fluorescent microscopy, you'll use a molecule called a fluorophore. So a fluorophore basically will be excited by a certain wavelength of light. It causes the electrons to jump. And then when the electrons fall back down, they release light at a different wavelength. So we call it excitation and emission. Different fluorophores have different emission wavelengths. So for example, these are images I actually took during my PhD with fluorescence microscopy. These are spirochetes. This is Borrelia burgdorferi. We did what's called a live dead stain. So cells that are alive are tagged with a green fluorophore. Cells that are dead are stained red. And so you can see the contrast here. Now, if we want to see things that are very, very small or want to have a really nice picture of the external structure of things like bacteria or even eukaryotic cells, we can use electron microscopy. So in this case, we are no longer using light to help us visualize our sample. Instead, electron microscopy uses electrons, a beam of electrons, to bounce off the sample, and then a computer takes that pattern and generates the image for you. So it's technically not a picture, it's called an electron micrograph. It relies on computational power to generate the image. It's very, very high resolution. And this is the only way that you can see viruses, but you can also see great detail on bacteria or even eukaryotes. You can see the cilia, the flagella, the fimbriae, all of these extracellular structures. So this is an image on the left. This is an electron micrograph from when I was a postdoc working for the Army. This is a biofilm of Staphylococcus aureus. So you see the coccus shape, and you can even see some of the extracellular structures that they've secreted. And then we can also see viral particles using electron microscopy. Larger organisms, of course, are going to be easier to see. So if a patient has a suspected infection with a eukaryotic agent like a fungus or another unicellular or even multicellular eukaryotic parasite, you can see those pretty easily. You often don't even need to stain, and you don't have to use a really high magnification on your light microscope. But if you suspect a patient has a bacterial infection, you're going to have to use the highest resolution light microscope you can, and you often will have to stain or prepare your samples in a certain way in order to more easily see those. As we talk about different types of infection, we'll talk about the different diagnostic techniques. But again, as people who have already taken microbiology, you should be very familiar with the different types of bacterial stains, including acid-fast, gram, endospore, simple, negative, and you should know what all of those will allow you to see. Okay. For some organisms, especially bacteria, some fungi, you may want to culture the organism. Again, this will depend on what you suspect the infection is. There are going to be some types of infection that are considered biosafety level 3. You, in most public health laboratories, will not have biosafety level 3 facilities, or you will not want the risk of culturing a biosafety level 3 organism. In Fresno County, we do have a number of different biosafety level 3 organisms that are found regularly or semi-regularly. These would include Coccidioides immitis, which is a fungus that causes valley fever. We will talk about that in our fungi section. Yersinia pestis, which is a bacterium that causes the plague, is found semi-regularly in the Sierra. Even a few years ago, they found it at Yosemite. We also have a bacterium called Francisella tularensis, which causes an infection called rabbit fever. We'll talk about that later this term. So there are some organisms you're not going to want to culture, but there are also a lot that you can at low risk to trained personnel. So there are lots of different types of media that you can use to culture organisms. Again, these terms should be familiar to you because you've taken microbiology. We have general or non-selective media that will grow most types of organisms, including blood agar, Mueller-Hinton, which is especially useful for doing MIC testing, antibiotic susceptibility testing. Thioglycolate broth, which is useful when culturing anaerobes. You'll probably remember the picture where you see the 5 different tubes, and there's bacteria at the bottom, the top, throughout, right? That can tell you the oxygen requirements of your organism. We can also selectively try to culture fungi using Saborodextrose agar. It tends to have higher sugar content and a little bit lower pH. So these are broad types of media that will grow a lot of different kinds of organisms. We also have selective, differential, and selective and differential media. Some of them are shown here. And then there are specialized types of media that grow only certain types of bacteria. For example, we could recover only E. coli, we could recover only Streptococcus, only Corynebacterium, only Vibrio. So these types of specialized media are useful when you have a good idea of what that infection might be. Now, some bacteria and some other types of organisms like viruses cannot be cultured on a plate. So especially with intracellular bacteria like Rickettsia, Chlamydia, Coxiella, these types of organisms are very, very difficult to culture because they won't grow on a plate. They will only grow inside of eukaryotic cells. So you'd have to be proficient in eukaryotic cell culture in order to grow some of these intracellular bacteria. You also have to be proficient in eukaryotic cell culture if you want to culture viruses, because viruses do require cell culture. These types of organisms can be much more difficult to isolate. So for intracellular bacterial infections or for viral infections, we often will rely on different techniques for diagnosis.